Hello everyone, this is Praveen from the Department of Chemical Engineering at Auburn University, going to present on my topic, which is titled as Co-Transport of Methanol and Carboxylate Ions in PEGDA-based cation exchange membranes. As we all know, the global carbon dioxide emissions is on the rise, and we need methods to reduce carbon dioxide and to prevent climate change. One such device which can be used to reduce carbon dioxide into various chemicals is called as CO2 reduction cell, which can reduce CO2 into various products such as formate, acetate, propionate, methanol, etc. To, to commercialize such a device, we need ion exchange membranes with sufficient ionic conductivities to protons uh, and while simultaneously mitigating the transport of the CO2 reduction products for its efficient operation. To be to bring such a device into reality, we can incorporate cation exchange membranes with negatively charged sulfonated group in its backbone, which can enhance the ionic conductivity of positively charged protons while uh, ensuring it can repel various CO2 reduction products such as formate, acetate, propionate, and methanol. So the figures here in the bottom show how the membranes in our labs are made. So the figure in the left shows the UV crosslinker which is used for the membrane preparation, and the figure on the right bottom here it shows how the membranes appear after its preparation. So to bring such a membrane into reality, we need to first prepare model cation exchange membranes with tunable crosslink densities and tunable monomer compositions. So we can incorporate PEGDA as a crosslinker with two different chain lengths and also tune the monomer compositions so that we can investigate how transport properties change or get affected by uh, various changes in the crosslinker chain length and the monomer compositions. So after preparing these membranes, we are subjecting these membranes to transport experiments such as permeability and solubility experiments. So in the permeability experiments, it is performed by incorporating a membrane in between two custom dual diffusion cells with pure water on one side and feed with 1 molar of solutes of interest such as methanol, propionate, formate, and acetate, as discussed previously. So we would like to track how much of these solutes gets transported through the membrane with respect to time, and the Yasuda's model helps us to calculate the permeability from this data. And the solubility experiments are also performed, which gives information on how much of the solutes get sorbed inside the membrane, uh, and this will help us in how the membranes uh, sorb the solute and desorb it into the other side of the cell. So till now we have performed the membrane permeability experiments for various solutes as discussed previously, and we have found that the permeabilities of formate ions are much higher than the permeabilities of acetate ions and propionate ions, and the results are consistent with two different cross-linker chain length. And we have also found that the permeabilities of carboxylate ions increase in co-permeation with fast-moving methanol molecules, as shown here in this middle two graphs. And we have also found that the water volume fraction increases with increasing SPMAC content, and the results are almost independent with varying cross-linker chain length. And in the future, we would like to perform membrane solubility experiments to formate acetate, propionate, and methanol, and also calculate the diffusivities of these solutes. At last, I would like to thank Auburn University and the DOE for their funding and the Buckingham Polymer Research Group for their help and support.